Chapter 3, Section 5, Catalysis and Enzymes. Now, when we go through and we do these calculations, the delta G, the delta E's, and all of that, that's all nice theoretical means of trying to discuss what's happening. Another mechanism for discussing what's happening in an enzymatic reaction is to go and look at what is referred to as the activation energy. How much energy has to be in the system for a reaction to occur on its own? Because that's the thing we got to remember about all these enzymatic reactions we'll be talking about over the next few chapters. All these reactions could and will occur on their own. It's just the time frame is going to not be biologically amenable. The reactions A plus B yields C plus D, that will occur on its own. Sometime in the next few minutes, few hours, few days, few weeks, few months, few years, it will occur. What is it waiting for? Enough activation energy to get in there to stress bonds. Waiting for A plus B, those two molecules to be in the right orientation to interact with each other, all sorts of things. But if you add in a catalyst, here a protein, that's going to take A plus B into its active site. It's going to orient them correctly. It's going to stress the bonds. And it's going to make this reaction occur with lower amounts of energy needed, lowered activation energy, and it's going to happen in minutes, seconds, milliseconds as its time frame. So we see here the red line. This is all by itself without an enzyme. All this energy has to be accrued into the system before you'll see A plus B go through any type of changes, any bond stress, any bonds getting ready to be reformed, for A and B to be in the correct orientation for things to occur. Add in an enzyme, and the amount of this activation energy gets greatly reduced. If we added in a third dimension here of time frame, time scale with the enzyme, things are logarithmically reduced timing-wise. Excuse me. So our enzymes are catalysts. They make things happen faster, more efficiently. We're going to see that enzymes typically are proteins. Not all of them. There are some that are RNAs or RNA-based, think our ribozymes. They are highly specific. One of the mantras you'll hear in all of my biology courses, even the biochemistry course I teach, is shape dictates function. The shape of the active site, where the work gets done in an enzyme, is of a certain shape that only certain shaped substrates or reactants can fit into it. Wrong shape, no function. So you'll see the reactants, also known as substrates. If you're discussing it as an enzymatic reaction, we refer to them as substrates. The substrate and the enzyme will interact at the molecular level. The R groups of the amino acids that make up the active site are interacting with the atoms that make up the molecules of the substrate. There is a direct interaction. There's hydrogen bonding, there's ionic bonding, there could be direct carbon-carbon, covalent bonds that are forming. So we see that there's always going to be some form of a cleft or whatnot that is where the substrate will be able to fit in. The only things that can fit into this active site and cause the enzyme to do its job, the cut, the paste, the twist, the tweak, it only does one, and only one thing. It turns out enzymes are pretty much one-trick ponies. Has to have the correct shape. The correct shape to fit in here to allow the R groups to be in the correct places to interact at the molecular level. Disaccharide goes in, cut, paste, twist, tweak. 2 monosaccharides are released. Enzymes reset, ready to go. As long as there's substrate there, it'll keep doing its job. Boom, 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 boom. Turns out, though, sometimes some of the enzymes are structured in such a way that they require a little helper known as either a prosthetic group or a coenzyme. The prosthetic group is actually part of the enzyme, the overall protein, where the activity will occur. The example here is heme in cytochrome. Cytochrome is a large complex of molecular complex, but it's the little heme molecule at the center that does the work. That's where you're going to find the interaction. Sometimes, though, the enzymes need to be paired up. Its little helper is going to be in the form of a coenzyme. Coenzymes, growing up, you heard these referred to as the vitamins. The reason why you have to have vitamins in your diet is because these interact with certain enzymes to get them to fold correctly so that they will have a functional active site. That's why people who are suffering from vitamin deficiencies will have metabolic deficiencies. They have the genes. The enzymes are there, it's just the enzymes are not in the correct shape. No shape, no function. And this is pretty much everything I just talked about. Everything has to be in the proper orientation, which the active site will hold the substrate in the correct orientation. There will be a complex. Things will interact at the molecular level. And it turns out that some of these enzymes can push through very endergonic reactions that have a high positive delta G, which means energy has to be poured into them. That reaction, so we see what is referred to as a coupling. Turns out these are going to be some of those reactions that require ATP. The ATP, that terminal third phosphate, the gamma phosphate, will be hydrolyzed, cut off, transferred either to the enzyme or to the substrate, and thus bringing in a negative delta G amount of exergonic free energy to power through this reaction.